The article, The Global Governance of Human Cloning, The Case of UNESCO, was written by Adele Langlois on the 21st of March 2017. UNESCO, also known as United Nations Educational, Scientific and Cultural Organization, set up a working group where they would investigate the possibility of legally binding convention to ban human cloning. Those in favor of reproductive cloning argue for liberal rights, citing reproductive freedom, or hope that cloning can tackle infertility. Others opposing will argue for the physical and psychological health of the clone. However, both sides agree that it is too risky based on the poor success rate in animal cloning. As an example would be Dolly. They took around 278 attempts to actually clone Dolly and create the first mammal, first cloned mammal. The health risks also apply to the mother and the child, which is why it's considered too risky. It was during a General Assembly where they had talked about this controversial topic and decided to settle on a declaration, as they could not resolve its members' differences regarding the reach of Convention on Human Cloning. However, it was not viable when it came down to the regulation of human cloning, because an international declaration, the United Nations Declaration on Human Cloning of 2005, already existed. Subsequently, the bioethics program went against these regulations and investigated the possibility of a convention on cloning in 2008. What is reproductive cloning? Reproductive cloning is the deliberate act of producing genetically identical individuals. J.B.S. Haldane is a British scientist who was the first person to have thought of the genetic basis for human cloning. In 2001, the first human embryos were cloned. However, they never grew beyond 6 cells. In that same year, the House of Representatives Standing Committee on Legal Constitution Affairs concluded that between reproductive cloning and stem cell research, it showed that there was more supporters shown towards stem cell research than there was to reproductive cloning. A year later, the federal government would look upon the issues for the research of stem cell, therapeutic cloning, and reproductive cloning and proved that legislations were required. In November 2007, Australia had banned human cloning and the creation of embryos for scientific purposes. However, in May 2007, the Infertility Treatment Amendment Act 2007 Victoria had passed a law which allowed somatic cell nuclear transfer in stem cell research. Who first realized objects could be cloned? The notion of cloning was first introduced in 1885 when a German scientist by the name Hans Driesch, who was a biologist and is known for his experimental work in the field of embryology. He began researching reproduction, and in 1902 he discovered a way to create a set of twin salamanders by dividing an embryo into 2 separate and viable embryos. This would later influence other scientists like John Gurdon in 1958, where he would clone frog skin cells of adult frogs. Many of these discoveries of cloning would be experimented through animals, as it would be too risky to produce hundreds of embryos. For example, Dolly the sheep, who on the 5th of July 1986 was known as the first mammal to be cloned from the cell of an adult animal, and it took scientists about 278 attempts to reproduce Dolly. However, there is yet to be any signs of progress, as in many countries such as Germany, Austria, France, and more have banned therapeutic and reproductive cloning. Though in 2006, the Australian Commonwealth Parliament passed a law which would allow the cloning of human embryos only for therapeutic purposes. And a year later, after the Infertility Treatment Amendment Act 2007 Vic had legalized the use of somatic cell nuclear transfer in stem cell research, moreover retaining the prohibition on human cloning for reproduction. And there are over 3 types of different artificial cloning, which include gene, reproductive, and therapeutic cloning.
The process of artificial human cloning may seem bizarre at first, but human cloning does occur naturally in the case of identical twins. Identical twins are individuals who developed from a single fertilized egg that had split, creating 2 or more embryos with almost identical DNA. But the chances of this occurring naturally is only 1 in 300 births. However, the topic at issue here is the process of artificial human reproductive cloning. Scientists are currently attempting to clone humans through the process of SCNT, or somatic cell nuclear transfer. This process involves the removal of a mature somatic or body cell from the selected donor animal, in this case a human, that is to be cloned. The DNA containing nucleus from the donor animal is then extracted from the nucleus of the somatic cell. This DNA is then injected using a syringe into an empty egg cell, also known as an oocyte, that has already had its nucleus removed. There is an alternative method to SCNT in which the entire somatic cell is fused with the empty egg cell using an electrical current. Once the egg cell develops into an embryo, it is then implanted into the womb of an adult female animal called the surrogate. One of the reasons why human cloning has not advanced beyond the development of a 6-celled embryo is due to the complexity of primate cells compared to the cells of other animals. The cloning process has to be done to a level of perfection that is unrealistic with the technology that we have at the moment, and failure to have that level of perfection would result in the embryo not surviving gestation, and even if it does survive, it would likely be born with a lot of diseases that would likely cause it to die within the first few days of its life. And one of the reasons for this is due to the spindle proteins. Spindle proteins are proteins that are essential for the process of cell division to take place. During metaphase of cell division, spindle proteins pull the chromosomes back and forth until they align along the equator of the cell. During anaphase, these spindle proteins separate the chromosomes and pull them to opposite poles of the cell. In animals such as rabbits, mice, and cats, the spindle proteins are located throughout the cell, so when you remove the nucleus, you aren't removing all of the spindle proteins. However, in primates, the spindle proteins are located in the nucleus around the chromosomes. So when you remove the nucleus, you are also removing the spindle proteins. So who will be affected the most by the discovery of human cloning? Recent studies show that 1 in 6 Australian couples are affected by infertility. Currently, there are options such as IVF and hormonal therapy for men that can be used to aid couples in having children. However, these options don't always work with all couples, so cloning provides an alternative to these options. Reproductive cloning is a complicated and time-consuming process that would undoubtedly cost a hefty price. Therefore, this option would likely be limited to those who are more socio-economically advantaged. Livestock reproductive cloning, on the other hand, is already helping farmers. Estimates place the number of cloned animals globally at around 6,000. This low number is likely due to the high cost of $15,000 USD to clone a cow and around $4,000 to clone a female pig. This is also probably due to the slow social acceptance of the topic. The cloning of pets is starting to become a trend in some companies, with companies willing to clone dogs and cats for around $50,000 using somatic cell nuclear transfer.
There are many ethical issues surrounding reproductive cloning. The sides supporting reproductive cloning argue that through understanding the composition of genes, it can allow scientists to make discoveries which could lead to genetic improvements in humans and help doctors cure diseases and disabilities. It would especially boost the research department when it comes to finding cures and learning more about a certain disease and how it evolves around its surroundings. Through human cloning, it could also lead to organ replacements, where vital organs such as the heart, the kidney, if they ever fail, they can be cloned and preserved as a backup for those in need. However, the opposing side feel that humans are special and that we were created in the image of God. Cloning would go against this belief and mean that the uniqueness of people will no longer exist. If there's only just one person, then that person would be so much more valuable, to those around them. The cloning success rate is low, and there is a higher risk of serious genetic issues or even shortened lifespans. It would stop the natural genetic improvements and differences, so then parents would likely have that option to pick what type of child they want. The creation of genetically enhanced humans will likely be exploited by others. At times, it could potentially be used to improve athletic performances or other factors to assist in a particular area. Health issues on human cloning. So health issues concerning human cloning would be that through this process it would lead to long-term defects and the process would result in higher failure rates, as it was apparent through the experiment when scientists cloned Dolly, who was the first mammal to be cloned from an adult animal. They had failed 277 times before finally succeeding after the 278th time. And from the results of a French scientist, also known as Dr. Jean-Paul Renard, he mentions that it can also lead to long-term health issues, and this is because he and his group had cloned a 2-month-old calf after taking genes from an adult cow. The 2-month-old would later die from blood and heart problems. In this experiment, it was reported that the cloning process had affected the normal genetic functioning of the developing calf. The cloned calf had been cloned from a skin cell which was extracted from the ear of a 15-day-old calf, and for the first month the clone was fine after being treated with diuretics for an enlarged heart. However, the rapid depletion of both the red and white blood cells had led its condition to deteriorate and would later die from severe anemia at the age of 7 weeks. Detrimental results would occur if these tests were tested on humans first, and hence is why we test on animals first.